Hello, in this video we will discuss about the cancer uncontrolled cell division. So the cancer is occur with a different gene segment, which that is basically the uncontrolled regulation, and there are many mutation will lead to cause cancer. Not a little mutation, because little mutation can be repaired, but the more damage of the DNA will lead to cause cancer, uncontrolled cell division. So that sometimes the regulate the cell multiplication breakdown. In this way, cell multiply and regulates more than the need of our body, and proliferate, differentiates an unwanted clone of cell called tumor formation. And this tumor formation is a basically group of cell production, and this group of cell will be uncontrolled and divide more and more due to the CDK and cyclin constriction more. So here is the somatic cell, which that is the person and contains somatic cell. The somatic cell is divided via a mitosis. So here I am drawing the cell cycle for understanding what is the mitosis and what is the cell cycle. So here is the cell. This is the normal cell, which that is going to the cell division. So the preparation first is the G0 phase, G1 phase. In this way, it can be G0 phase without unmitotic phase. But here is G1 phase is the growth 1 phase. In this way, the growth 1 phase, the chromosome decondensation, reformation of nuclear envelope, and cytokinesis will be occur. In this way, the cytokinesis will be occur from this going to the envelope, and cytokinesis means the site or mean the cell will be occur. And after this, karyokinesis will be occur, the karyokinesis. So in this way, here you can see this is the DNA synthesis, and this is after the DNA synthesis is known as the S phase. And after the S phase, the chromatids will be double. So the double chromatid is for the replication and divide the cell into 1 into 2. So these two cell divide, and in this way the daughter cell will be produced from one cell to the two cell, and both contain a diploid. The number of chromosome will remain constant. So in this way, here you can see the chromosome segregation, nuclear envelope breakdown, and chromosome condensation formation. And in this way, the cytokinesis will be occur after the karyokinesis. So the karyokinesis to the cytokinesis to divide and daughter cell. And some cell, for example, the nervous cell and hepatic cell or pancreatic cell, will go into the G0 phase, which that is the undividable, and that is specialized. So here is the CDK and cyclin. Cyclin dependent kinase, and cyclin will bind with a protein which that is transcribed from the DNA, and that is the constriction of the cyclin and CDK in each phase is regulate. So cycling up and down will maintain the cell division, while the increase of the here is the mitosis and apoptosis will be balanced. If the cyclin and CDK will be decreased, so the apoptosis will be occur, while the cyclin and CDK will be increased, so the mitosis will be occur. So here is a tumor will be two type, and if the uncontrolled cell division via a mitosis is known as the tumor formation, and benign tumor it is as well as malignant tumor. Benign tumor is not harm and look like normal cell group and secrete substance, while the malignant tumor or malignancy is the divide more rapid and proliferation uncontrolled, and mostly invade surrounding tissue like pancreas, liver, digestive tract, etc. So go into the circulation and metastasis stage, which that is the uncurable and establishment to another organ. So let's begin. Here is the mechanism and stages of the cancer causing and proliferation. So here is a normal cell, zero phase, which that will be ready after the damage of the DNA, and the growth factor will stimulate this. one phase and selective phase. In this way, the cell will be select for the proliferation due to the damage of the DNA, and the phase 2 will be proliferate, and phase 3 is basically going to the metastasis after the fourth stage. And this proliferation of the cell will be smaller than the normal cell because the nucleus will be remain constant, but the cytoplasm will be not more volume due to the rapid cell division. So the risk factor we will discuss here is basically the UV radiation, chemicals, epigenetics, and aging, lifestyle, air pollution. So these all things is the risk factor for the cancer. So important things, let's begin to recap one by one for understanding what is the risk factors. So first of all, we will understand about that UV radiation. UV radiation is basically the sunlight which contain ultraviolet radiation, which that will be damage the DNA. After the damaging of the DNA, the mutation will be caused, and the oncogene suppressor can be damaged. So in this way, the oncogene uncontrolled transcription to production of protein and oncoprotein. This oncoprotein is lead to due to the point mutation, deletion and addition of the nitrogenous base, and in this way the point mutation will lead to cause the cancer due to the more and more protein synthesis, which that is related to the oncogene, and cell proliferation and cell growth will be more. So here is the chemical. Chemical can be chemotoxin or other things, which that is an alcohol or medicine. So in this way you can see colorectal cancer, colon cancer, and chemotoxin will damage the DNA, such as alcohol will lead to cause cancer. Also the when chemicals absorb effect to other organs. So in this way here you can see the colorectal cancer caused due to the chemotoxins and medication, which that is trigger the DNA changes and mutation, epigenetic changes, which that is basically also related to the epigenetic. So it will lead to cause cancer due to the mutation of DNA. And here is the epigenetic. Epigenetic means the CPG island on promoter region are present, which that is pressed the after the methylation will not express for oncogene stimulation. So when oncogene stimulation after the acylation or activation of the promoter region lead to gene expression due to demethylation of promoter region of the gene, or also on the suppressive genome methylation. So that is important. And here is the aging and lifestyle will totally depend to the food, and basically aging will cause cancer if we will take food badly and not fasting. So the medication and water, stress, and that is inflammation, fasting, and fasting less and eating more will lead to cause aging, and aging is the basically the major reason for the cancer. And lifestyle changes, for example, not healthy food but unhealthy food consumption. While here is the air pollution is the 90% cancer causing agent. Basically the oxygen and carbon dioxide exhale and inhale, which that is macrophage are present is here, and smoking will lead to cause the air pollution will directly contact by inhalation to cause damage epithelial cell. Exposure of some smoke and other toxins inhalation, which that will lead to cause cancer because the toxins are present. For example, the smoke in the cigarette, which that lead to cause the cancer due to there are many toxins are present. So here is the cells. Basically the cell is proliferation during the growth factor, for example insulin, as well as the growth somatostatin hormone, which that is released from the pancreas, as well as adenohypophysis of the pituitary gland will lead to also its contain growth hormone, insulin like growth factor 1 and 2. So let's begin to understand. Here is the bigger picture for understanding the mechanism of cancer, and here is just for understanding the cellular mechanism and the molecular pathway. So here I am drawing the plasma membrane. This is a plasma membrane. This plasma membrane is made up of phospholipid, wide layer of phospholipid. Here is the extracellular matrix, extracellular space, and intracellular space. So here is the insulin-like growth factor binding protein will bind and will not allow to rebind, but the insulin-like growth factor binding protease enzyme used to debindation or debinding to release the IGF-1 and will bind with the tyrosine kinase receptor. Insulin-like growth factor 1 receptor basically is the intracellular domain and extracellular domain, which after the binding to phosphorylation of the tyrosine kinase to cascade of different phosphorylation of the different protein, that is the cytosolic protein, and the plasma membrane binding protein RAS. RAS is the inactive during the GDP contain, and the phosphoinositide 3 kinase, which that is bind and activate, and JAK JNS kinase pathway will be bind. JAK pathway will be bind and stimulate after the phosphorylation, and the RAS will contain RAF. This RAF is activated after the activation of the RAS by GTP, and the RAF will be phosphorylase to AMPK and activated mitogen AMPKKK kinase pathway, which that will lead to stimulate and phosphorylation of the RAF to MUC mitogen activated protein kinase, and after the ERK protein kinase, which that will lead to transcriptional factor becoming the transcriptional factor. While here the PI3K pathway, which that activates the AKT, and AKT will lead to major the stimulation of the mTOR pathway, and rapamycin will inhibit it. While the mTOR pathway is the autophagy inhibition and apoptosis inhibition, because the autophagy is the directly linked with the apoptosis. So here is the mTOR will trigger the BCL2 and BAD protein. This BCL2 and BAD will bind with the DNA in the nucleus is here. Here is the chromosome which contain DNA. So remember about that. This is the rapamycin is the major target for the basically the mTOR pathway inhibitor. While the mTOR will lead to cause cancer due to the proliferation, while the inhibition of autophagy. So the BCL2 and BAD will stimulate by the transcriptional factor, and ERK will bind with the transcriptional factor to growth regulatory gene will basically stimulate to proliferation of the cell and growth of the cell, and BCL2 and BAD and BCL2, which that will lead to binding the polymerase enzyme to lead the P7056K protein K gene, and 4EB D1 protein, which that is lead to transcription, and in this way transcription to become a proliferation of the cell. Same mechanism, you should know about that. And here is the E2F gene, which that lead to Fox M1 gene activation, and in this way the proliferation basically the separately it will be transcribed, not differently. So E2F and Fox M1, which that is lead to proliferation. And here is the mice and rats, which that is the here is the repressor. So the repressor will repress while during the growth repressor segment, while it will lead to cell proliferation after the mice and rats activation, and genes produces cyclin and CDK more than the need. So here the messenger RNA will be produced from the different mechanism, and in this way after the binding with the transcriptional factor to produce the messenger RNA with the desired messenger RNA on the basis of the growth factor uh, uh, binding stimulation to cell division, invasiveness, mitosis, meiosis, proliferation, apoptosis decrease, and cell survival will be increased, and development of cancer cells will be occur. And in this way you should know about that. Here is the STAT3, which that is stimulated by the JAK, and in this way it is also the transcriptional factor. And in this way I hope you make sense about these things, the cancer, uncontrolled cell division, the molecular pathway, as well as the overview what is that. So if you have understood, please comment below and give the feedback. Thanks for watching. Please subscribe my channel.